Okay, so first of all, thank you for the kind introduction and thanks to everyone for joining. So in this presentation, I would like to discuss our recently published analysis of the blood metabolome in Parkinson's disease, which we conducted as part of a national cohort study here in Luxembourg. To give you a short outline of the talk, I will start with a brief background on Parkinson's disease, the cohort and our metabolomic study design. Then in the first analytical part, I will discuss the differential analysis of metabolites for untreated de novo patients, treated patients, and controls. I will then move on to the more global perspective of pathways and network levels. And finally, I will present a predictive machine learning analysis of the metabolite data. So since not everyone in the audience may be familiar with Parkinson's disease, I just want to give a one-slide background on the disease. Parkinson's is a currently incurable brain disorder. There are symptomatic treatments available, but no disease-modifying therapies. And the symptomatic treatments tend to lose efficacy over time and are associated with significant adverse effects. The disease affects mainly people over 60 years of age, and less commonly younger people. And we can distinguish genetic and sporadic forms of the disease, which also differ in terms of disease progression and manifestations. The most characteristic cardinal disease symptoms are motor impairments, including resting tremor, rigidity, slowness of movement, and postural instability. However, there are also several less non-specific motor impairments, such as sleep disorders, constipation, and sensory impairments, such as loss of smell. And some of these non-motor symptoms can also occur early in the prodromal phase of the disease, many years before the onset of cardinal symptoms. In general, the symptoms are very heterogeneous. And here on the bottom right, you see two famous patients who reflect both the heterogeneity of the disease course and the manifestations. The actor Michael J. Fox on the left developed the disease at a young age when he was 29, likely also due to genetic influences, and it progressed rather slowly, whereas the boxer Muhammad Ali on the right developed the disease in his 40s, probably influenced by boxing-induced head trauma, and he experienced a rather rapid decline. So this disease has many different phases, and our aim is to understand it better. For this purpose, the Luxembourg Parkinson study was initiated around 10 years ago. It is a nationwide monocentric observational longitudinal study, and currently the cohort covers around 800 patients with typical or atypical forms of Parkinsonism and a similar number of controls. The patients undergo an annual follow-up, while the controls are followed up only after 4 years. The collected clinical data includes standard assessments of motor symptoms using the MBS-UPDRS rating scale, but also assessments of several non-motor symptoms, including cognition, sleep, quality of life, environmental factors, among others. Furthermore, biosampling is conducted to obtain biospecimen from several body fluids and tissues, as you can see on the bottom left, and we use these biosamples for molecular profiling analysis to better understand the changes in the disease. One of our main molecular profiling analysis of the cohort was the LuxPark Metabolomics study, which we conducted together with Metabolon. This study had two main goals. First, we wanted to characterize blood metabolomics changes in Parkinson's versus controls, as well as in distinct Parkinson subgroups. Second, we wanted to interpret cellular pathway and network alterations and build prediction models for clinical outcomes. The data was obtained by applying the LCMS metabolomics profiling approach by Metabolon, using 1,500 blood plasma samples from the cohort and covering measurements for around 1,400 metabolites. As shown on the bottom left, these 1,500 samples included a baseline analysis of 1,300 samples, comparing the metabolite profiles of 593 Parkinson patients, including also a small subset of untreated de novo patients, and a similar number of controls. In addition, we investigated a few special subgroups, including Parkinson patients with dementia and patients with atypical forms of Parkinsonism. Moreover, as shown on the bottom right, we also performed a longitudinal analysis for a smaller subset of 100 patients included in the baseline, covering measurements for 2 consecutive annual follow-ups with a balanced representation of the sexes. Before discussing the methods and results, I would like to mention a few important study limitations upfront. First, regarding dietary effects, there was no possibility to ensure that all subjects enrolled in the study received the same diet or participate in the fasting state. So therefore, we need to consider dietary influences as part of the data interpretation. Second, we needed to normalize for batch effects. So the samples in the study were allocated across multiple measurement batches, and only an approximate balancing of sample group representations was possible. So therefore, we have to expect an increased data variance here. However, we can to some extent compensate this by the normalization and by a large sample size. Finally, we also have strong drug treatment effects in our data because most patients received dopaminergic treatment, which can confound the measurements. So therefore, we have adjusted the analysis for confounding effects, and we also compared the treated patients against a subset of untreated de novo patients. An important component of the study design was an algorithmic sample matching and collection, which we conducted before the study to increase the statistical power. Our goal was to select baseline biospecimens from patients and controls in our biobank, such that the sexes are matched and balanced, the age and body mass index distributions are matched, the use of arterial medication is balanced between patients and controls, and we minimize the occurrence of other comorbidities, in particular comorbidities treated with non-PD related medications that can confound the measurements. The two main benefits of this algorithmic sample selection are that it reduces the influence of confounding factors, and it helps us to remove unwanted sources of variance in the data. As a result of this algorithmic selection, we could ensure that the sex representations were matched between patients and controls, as you can see here on the top. We also observed a similar percentage of individuals taking arterial medication among the patients and controls, as well as roughly similar percentages of subjects taking medications for other symptoms. Furthermore, as you can see in the density plots below, the age and body mass index distributions were highly similar between patients and controls. So we can really limit here the impact of several common confounding factors. Finally, we also performed several quality controls to check for outlier samples and potential biases in the data distribution, and these analyses confirmed the high quality of the data. As a first analytical approach, we performed a differential statistical analysis. Specifically, our goal was to identify only those PD-associated metabolite changes that are independent from medication, age, and sex effects. And for this purpose, we compared Parkinson patients versus controls using the empirical-based moderated t-statistic. This test is robust against deviations from a normal distribution, and it can increase statistical power by shrinking the metabolite-wise variance estimates towards the global mean. Importantly, the analysis was adjusted for confounding factors, including age and sex as standard confounders, but also dopaminergic treatment effects that represented a particular challenge for this data. To capture these effects of dopaminergic medication, we used the metabolite measurements of one of the most important L-DOPA drug metabolites, namely 3-O-methyl-DOPA, as a confounding variable in our model. In addition, we compared the statistical results for treated patients with those for the subset of untreated de novo patients to ensure that we obtain reliable treatment-independent results. Finally, we checked for other possible confounders in our clinical data by performing a principal variance component analysis, and this analysis did not suggest the presence of any additional confounders with a major effect in our data. So here you see a volcano plot representation of the results for de novo PD versus control, and this shows the metabolites that differ the most between untreated patients and controls, ensuring the results are independent from dopaminergic treatment effects. The horizontal axis shows the effect size in terms of the log4 change, and the vertical axis the significance in terms of the negative decadic logarithm of the p-value. So basically, higher values are better. And in this plot, the green points represent all metabolites that had both high significance and high effect size. The red points, those that had only high significance. The orange points, those with only high effect size. And the remaining metabolites are shown in gray. So firstly, we see here that we have several metabolites with high significance and also pronounced effect sizes. And secondly, we observed that certain types of metabolites were overrepresented among the significant findings. In particular, as indicated by the black lines on the right, we noticed that multiple top-rank metabolites are involved in the xanthine metabolism pathway, and I will further discuss this on the next slides. So first here you see a more quantitative representation of the statistics for the top-ranked metabolites for the de novo PD versus control comparison. And I have highlighted here in blue the xanthine metabolites, which are all statistically significant and all have a higher abundance in patients compared to controls. As you can see, apart from the xanthines, there were also several other classes of metabolites showing significant alterations. But in terms of metabolite groups belonging to the same functional class or pathway, the xanthines clearly stood out in this ranking. Therefore, the xanthines also became a key focus of our study and the further investigations we performed. After studying the untreated de novo patients, we also compared the treated patients versus controls, adjusting for confounding effects. So here you see the corresponding volcano plot of the results. And although we had adjusted the analysis for dopaminergic treatment effects, we noticed that multiple of the top-ranked metabolites here shown at the top are well known to be associated with dopaminergic treatment. As we did not see these changes in the de novo patient versus control comparison, we believe that these are still remaining treatment effects and that the confounder adjustment cannot fully remove all the treatment effects from the data. And so therefore, as a next step, in order to identify those significant metabolites that are supported both by a large sample size, but that are also clearly independent of treatment effects, we compared and intersected the results from the analysis of treated and untreated patients. So here you see the shared significant metabolites that are both probably significant for the untreated de novo patients on the left and for the complete cohort involving the treated patients on the right. So where we can be sure that these results are well supported across many subjects and are also clearly treatment independent. And again, also in this filtered ranking, we see some of the most significant and strongest changes among xanthine metabolites. In particular, the two top-ranked metabolites highlighted in blue belong also to this group. Next, we moved from the analysis of single metabolites to the more global perspective of cellular pathways enriched in multiple metabolite changes using the KEC database as a pathway resource. And while a number of different pathways showed interesting coordinated changes in the data, we noticed that multiple of the top-ranked pathways were enriched in xanthines, such as the purine metabolism pathway highlighted here, which I will focus on in the next slide. So if you look at the core interactions in the purine metabolism pathway, shown here in the center, we can see that they link all four of the top altered metabolites in our data together by a small set of enzymatic reactions. On the left and right corners, you can also see the box plot representations of the xanthine changes in de novo PD versus controls, highlighting that all four metabolites in this pathway are significantly increased in our data. To better understand these results, we investigated prior functional links between the xanthines and Parkinson's disease in the literature. And interestingly, xanthine derivatives have already been proposed as drug targets for Parkinson's because their administration showed benefits in both human Parkinson patients and mouse models such as neurotoxin-based models for neurodegeneration. Here below, you see the structures of common xanthine derivatives found in humans. And one of these derivatives is actually caffeine, shown in the center, which has also been associated with protective effects in Parkinson's disease. However, this is unlikely due to a generic stimulant effect, as previous investigations on the general use of stimulants in Parkinson's found that they were mostly not protective in the disease. The xanthine structures are also used often as scaffolds in various drugs, including the FDA-approved Parkinson drug istradefylline, which is an adenosine A2 receptor antagonist. And indeed, one of the proposed mechanisms for the beneficial effect of xanthines in PD is A2A receptor antagonism, because this is known to enhance dopaminergic signaling and may therefore help to counteract the loss of dopaminergic neurons in Parkinson's disease. However, this is not the only mechanism suggested in the literature. Another one is the inhibition of the enzyme monoamine oxidase, or MAO-B, which is known to reduce dopamine breakdown and is therefore also a well-established drug target used for many years in Parkinson's treatment. A further previously proposed link between xanthines and Parkinson is the generation of reactive oxygen species by one of the main enzymes in xanthine metabolism, namely the enzyme xanthine dehydrogenase, or XDH. So XDH converts hypoxanthine to xanthine So these are two of the top significant altered metabolites in our dataset. And this conversion, as shown on the right, is known to produce reactive oxygen species. These reactive oxygen species in turn induce oxidative stress, which can damage mitochondria and neurons in Parkinson's disease. Furthermore, increased levels of XDH have previously been observed in Parkinson patients, and this supports basically their potential involvement as an important mechanism in the disease. In general, in Parkinson's disease, oxidative stress plays an important role because it contributes to the loss of dopaminergic neurons in the midbrain. The relevance of oxidative stress is also supported by other observations in our data. For example, a decrease of gamma-glutamyl-alanine in PD, which we observed in the metabolite data obtained here, and which may affect glutathione metabolism and reduce the antioxidant defense. In order to better understand the mechanisms and effects of xanthine changes in our data, as a next step, we performed an integrated metabolomics and transcriptomics network analysis. In particular, we complemented the metabolomics data by the analysis of both blood and brain transcriptomics data from Parkinson case control studies to identify enzymes with expression changes that are associated with significant metabolites in our data. And indeed, we identified one enzyme, hypoxanthine phosphoribosyl transferase, or HPRT1, shown here in the top center, which is significantly decreased in Parkinson's in both blood and brain samples, and which plays a central role in xanthine metabolism. As you can see in the metabolic subnetwork shown here, HPRT1 converts hypoxanthine and phosphoribose diphosphate into inosine monophosphate and pyrophosphate. Now, inosine monophosphate, or IMP, as you can see here on the right, is an important precursor of ATP, and therefore reductions in HPRT1 activity have previously been associated with cellular ATP deficiency. Thus, we believe that the decrease in HPRT1 levels may be the main mechanism that explains the accumulation of xanthines observed in the left branch of this pathway, and potentially also other pathological downstream changes in Parkinson's disease. Interestingly, a genetic HPRT1 deficiency called Lesch-Nyhan syndrome involves loss of dopamine in the basal ganglia and hyperkinetic movements, which is reminiscent of Parkinson's disease. Moreover, HPRT1 overexpression was tested as an intervention in a mouse model of neurodegeneration, the 6-hydroxydopamine model, and this inhibited dopaminergic neuron loss by activating beta-catenin signaling. The susceptibility of this pathway for pharmacological modulation was also indicated by a previous study in human patients. Specifically, in an open-label, single-arm clinical trial with 26 Parkinson patients, an intervention was tested in the left branch of the pathway you see here above, giving patients both the xanthine inosine and an inhibitor of the enzyme XDH. The authors could show that this treatment had multiple beneficial effects. It increased blood hypoxanthine levels and cellular ATP, and it decreased motor impairments in the patients as measured by the UPDRS-3 score. So overall, in contrast to the potential mechanisms discussed on the previous slides, this mechanism here, linking alterations in xanthines with the enzyme HPRT1 and with cellular ATP deficiency in Parkinson's, is the one that receives the strongest support from our data. I mentioned at the beginning that we also collected longitudinal metabolomics data over 2 annual follow-ups for a subset of 100 patients in our cohort. Accordingly, as a further analysis, we also checked how xanthine metabolites change over time. As you can see in the box plots here, in 3 out of 4 of the xanthines that were significant in the cross-sectional analysis, we also observed significant increased abundances over time. The effect sizes were small, but the consistent and significant results indicate that there are also relevant longitudinal changes in xanthine metabolism in PD, which further corroborates the relevance of this pathway in the disease. Finally, we conducted a machine learning analysis of the data to see whether individual metabolites have predictive power for de novo PD versus control classification. The focus here was on ranking individual features rather than on building a more complex multivariate model. Specifically, we tested each metabolite individually as a predictor using both linear and radial support vector machines to predict the diagnostic outcome. And then we measured the predictive performance using the area under the receiver operating characteristic curve on both training and test set partitions of the metabolites data. While multiple metabolites showed a modest predictivity, as you can see here, the metabolite xanthine achieved the highest average area under the curve, consistent with our previous statistical differential abundance analysis. This machine learning investigation is of course not independent from the previous statistical analysis because we used the same data, but it indicates that the pronounced differences in xanthine levels between patients and controls might also serve as a potential predictive feature in biomarker models of the disease. So to conclude, I just want to give a final summary of the main results across these different analysis types, and then basically summarize what we obtained as outcome. So in the metabolomics, in the metabolite level analysis, we identified statistically significant alterations in de novo patients versus controls, particularly in multiple xanthines. When we studied the metabolite changes at the pathway and network level, we confirmed coordinated Parkinson-associated changes in purine and xanthine metabolism. And an integrated analysis with transcriptomics data suggested the enzyme HPRT1 as a key regulator of these changes. Finally, in the machine learning analysis, we identified candidate metabolites with modest significant predictivity for de novo patient versus control discrimination. And xanthine is the top-ranked predictor reflecting the pronounced alterations of this metabolite. Yeah, finally, I'd like to thank the members of our team shown here who are behind the work I showed you, and I thank you for your attention.